Welcome to our countdown of the top 10 creepiest creatures under the microscope. Prepare to be amazed and a little bit creeped out by these tiny terrors. In the vast unseen tapestry of life that surrounds us, a universe teems with organisms so minute they defy the naked eye. Yet when magnified, these hidden realms reveal beings of extraordinary complexity, sometimes beautiful, often bizarre, and occasionally profoundly unsettling. Today we embark on a journey into this Lilliputian world, a safari through droplets of water, specks of dust, and even the surfaces of our own bodies. Here, the familiar rules of scale and form are distorted, giving rise to creatures that might seem plucked from the pages of science fiction. Their survival strategies, their intricate anatomies, and their often parasitic lifestyles offer a glimpse into the relentless ingenuity of evolution. These are not monsters from myth, but real living entities playing out their dramas in a theatre invisible to us until now. So adjust your focus and steady your nerves as we delve into the lives of these microscopic marvels, each with a story to tell, a story that might just change the way you see the world. We shall explore creatures that can withstand the vacuum of space, others that call our very skin their home, and some whose existence is a delicate dance of predation and parasitism. This is a world where a single cell can be a formidable hunter, where beauty and horror often intertwine, and where the line between the minuscule and the mighty blurs. Join me, David, as we peel back the veil of the visible and confront the fascinating and sometimes unnerving inhabitants of the microcosmos. Prepare to witness life in its most astonishing and at times creepiest detail. Our first subject, the tardigrade, often affectionately known as the water bear or moss piglet, is a creature that redefines our understanding of resilience. These microscopic invertebrates, typically less than a millimeter in length, inhabit an astonishing array of environments, from the highest mountain peaks shrouded in icy mists to the deepest sea trenches where sunlight dare not penetrate. They are found in lush mosses and lichens, damp soils, and even in the ephemeral puddles that form after a rain shower. Their ubiquity is a testament to their extraordinary adaptability, a quality that makes them a source of endless fascination for scientists. Under the microscope, the tardigrade presents a rather endearing, if somewhat alien, appearance. It possesses a segmented body, stout and somewhat cylindrical, supported by 4 pairs of stubby legs, each tipped with sharp claws. These claws are not for aggression, but for clinging steadfastly to their substrate, navigating the miniature landscapes they call home. Their movements are slow, deliberate, almost ponderous, giving them their bear-like moniker. They trundle through their watery world with an unhurried gait, seemingly unperturbed by the giants, us who might be observing their every move from above. What truly sets the tardigrade apart, and perhaps lends it a certain creepy resilience, is its ability to enter a state of cryptobiosis. When faced with environmental conditions that would prove lethal to almost any other life form, extreme desiccation, freezing temperatures, intense radiation, or even the vacuum of space, the tardigrade can suspend its metabolism almost entirely. It retracts its legs, expels nearly all the water from its body, and transforms into a tiny, desiccated husk known as a ton. In this state, it can endure for decades, patiently waiting for conditions to improve. Imagine, if you will, an organism that can survive being boiled, frozen to near absolute zero, and bombarded with radiation levels 1,000 times higher than what a human could withstand. This is the reality of the tardigrade. When moisture returns, the toon rehydrates, and the creature slowly, miraculously returns to life, resuming its activities as if merely waking from a long slumber. This capacity for suspended animation, for cheating death in the face of unimaginable adversity, is both awe-inspiring and, in its sheer tenacity, a little unsettling to contemplate. Their diet consists primarily of plant cells, algae, and sometimes even smaller microorganisms, which they pierce with sharp stylets in their mouths, sucking out the nutritious contents. Despite their incredible toughness, They play a humble role in their ecosystems, contributing to the decomposition of organic matter and serving as a food source for slightly larger microscopic predators. The tardigrade, then, is not just a curiosity, it is a profound example of life's sheer will to persist. A tiny titan that thrives where others perish, its existence a constant reminder of the hidden strengths within the smallest of packages. Next, we turn our gaze to a creature that lives quite literally under our noses and indeed all over our faces. This is the Demode mite, a microscopic arachnid, a distant relative of spiders and ticks that makes its home in the hair follicles and sebaceous glands of mammals, including a vast majority of human beings. Though the thought of tiny, eight-legged creatures crawling on our skin might induce a shiver, these mites are, for the most part, harmless commensals coexisting with us in a relationship that has evolved over millennia. Two species are commonly found on humans, which resides in hair follicles and which prefers the oil-producing sebaceous glands. Viewed under high magnification, the Demode mite has an elongated, almost worm-like body, perfectly adapted for navigating the narrow confines of a hair follicle. Its 8 short, stumpy legs are clustered near its head, equipped with tiny claws for gripping onto hairs. Its mouthparts are designed for consuming skin cells and sebum, the oily substance our glands produce to keep skin and hair lubricated. They are largely nocturnal, emerging from their follicular homes under the cover of darkness to mate on the surface of our skin before retreating once more. This clandestine lifestyle means we are usually entirely unaware of their presence. The life cycle of a Demode mite is intimately tied to its human host. Females lay their eggs within the hair follicles or sebaceous glands. These hatch into larvae, which then develop through several nymphal stages before maturing into adults. The entire process from egg to adult can take a couple of weeks. While they're generally benign, in some individuals, particularly those with compromised immune systems or certain skin conditions like rosacea, an overpopulation of Demode mites can be associated with inflammation and irritation, leading to itching and redness. However, for most of us, they are simply silent, unseen passengers. The creepy factor associated with Demode mites stems largely from their ubiquity and their intimate proximity to us. The knowledge that our eyelashes, eyebrows and facial skin are teeming with these microscopic arachnids can be disconcerting. They are a constant, invisible reminder of the complex ecosystem that our bodies support. It is a humbling thought that we are not solitary beings, but walking habitats for a multitude of other organisms. Their existence challenges our notions of personal space and cleanliness, even though their presence is a natural part of human biology. Despite their somewhat unsettling reputation, Demode mites play a role, albeit a small one, in the ecology of our skin. By consuming dead skin cells and excess sebum, they may even contribute in a minor way to skin health. Understanding these tiny inhabitants is not just about confronting an uncomfortable truth. It's about appreciating the intricate web of life that extends even to the surface of our own bodies. They are a testament to the pervasiveness of life, finding niches in the most unexpected of places, including the very skin we live in. We now venture into the world of the amoeba, a name that often conjures images of a simple primordial blob. Yet this single-celled organism is a marvel of minimalist design and predatory prowess. Amoebas belong to a diverse group of protists, characterized by their ability to alter their shape, flowing and oozing their way through their aquatic environments. They lack a fixed cell wall, allowing their cytoplasm to extend outwards in temporary projections called pseudopodia or false feet. These pseudopods are not just for locomotion, they are also the amoeba's primary tools for hunting and engulfing prey. Observing an amoeba under the microscope is like watching a slow, deliberate dance of life. The cytoplasm streams within the cell membrane, pushing outwards to form a pseudopod in one direction while the rest of the cell body follows. It moves with a creeping, almost ghostly motion, exploring its surroundings for sustenance. When it encounters a food particle, perhaps a bacterium, an alga, or another smaller protist, the amoeba extends its pseudopods to surround the prey, eventually enclosing it within a food vacuole inside its cell. This process, known as phagocytosis, is a fundamental mechanism of feeding in many simple organisms. The internal structure of an amoeba, though lacking complex organs, is a hive of activity. A distinct nucleus houses its genetic material, controlling the cell's functions and reproduction. Contractile vacuoles rhythmically pump out excess water, maintaining the cell's osmotic balance, particularly crucial for freshwater species. Food vacuoles containing ingested prey drift through the cytoplasm as digestive enzymes break down the meal. Despite their apparent simplicity, amoebas are highly efficient, self-contained living units, capable of all essential life processes within the confines of a single membrane. The creepy aspect of the amoeba, for some, might lie in its amorphous, ever-changing form. It lacks the defined structure we associate with most living creatures, appearing almost like a sentient droplet of jelly. Its ability to silently stalk and engulf other microorganisms can also seem somewhat predatory and relentless on a microscopic scale. Furthermore, while most amoebas are harmless, free-living organisms found in soil and water, a few species can be pathogenic to humans, causing diseases like amoebic dysentery, a stark reminder that even the smallest of creatures can have a significant impact. Amoebas reproduce asexually, typically through binary fission, where the cell divides into two identical daughter cells. This simple yet effective method allows for rapid population growth when conditions are favorable. Their adaptability and diverse feeding habits have enabled them to colonize a vast range of habitats, from freshwater ponds and stagnant pools to moist soil and even the intestines of animals. The amoeba, in its fluid form and predatory nature, represents a fundamental and enduring form of life, a shape-shifting hunter navigating the microscopic wilderness with silent, flowing grace. Chapter 4. The Teardrop Terror, Giardia's Hidden World. Our next microscopic subject is Giardia lamblia, a flagellated protozoan parasite that is notorious for causing a debilitating diarrheal illness known as giardiasis or beaver fever. This tiny organism, often found in contaminated water sources around the globe, presents a surprisingly intricate and almost symmetrical appearance when viewed under a microscope. An appearance that belies its capacity to cause considerable discomfort to its hosts, including humans, dogs, and yes, beavers. Its distinctive teardrop or pear shape, coupled with multiple flagella, gives it a somewhat alien yet strangely organized look. The Giardia trophozoite, the active feeding stage of the parasite, possesses two nuclei, giving it a somewhat face-like appearance with two dark eyes. It also has a ventral adhesive disc, a specialized structure that allows it to attach firmly to the lining of the small intestine of its host. This tenacious grip is crucial for its survival and proliferation within the gut. 8 flagella, whip-like appendages, trail from its body, providing motility and enabling it to navigate the intestinal environment. This combination of features makes it perfectly adapted for its parasitic lifestyle. The life cycle of Giardia is a tale of two forms. Outside the host, it exists as a hardy cyst, an environmentally resistant stage that can survive for weeks or even months in cold water. These cysts are shed in the feces of infected individuals or animals. If ingested by a new host, typically through contaminated food or water or via the fecal-oral route, the cyst undergoes excistation in the small intestine, releasing 2 trophozoites. These trophozoites then multiply by binary fission, colonizing the intestinal surface and interfering with nutrient absorption, leading to the characteristic symptoms of giardiasis. The creepy nature of Giardia lies not only in its parasitic existence within our bodies, but also in its stealth and resilience. The cysts are invisible to the naked eye, lurking in seemingly pristine mountain streams or inadequately treated water supplies. The symptoms of infection, diarrhea, abdominal cramps, bloating, nausea and fatigue can be prolonged and severe, significantly impacting an individual's quality of life. The thought of these microscopic face-like organisms clinging to one's intestinal walls and disrupting bodily functions is undeniably unsettling. Preventing giardiasis hinges on good hygiene practices and ensuring the safety of drinking water, particularly in areas where sanitation may be compromised or when venturing into wilderness environments. Boiling or filtering water effectively eliminates Giardia cysts. Despite its unpleasant effects, understanding Giardia's biology and life cycle is crucial for developing effective treatments and control measures. This teardrop-shaped parasite, with its eerie symmetry, serves as a potent reminder of the invisible threats that can lurk in the most idyllic of natural settings, a microscopic invader with a significant impact on global health. The malaria harbinger Plasmodium's deadly dance. We now confront Plasmodium, a genus of unicellular eukaryotes that are obligate parasites of vertebrates and insects. Among these, several species are infamous as the causative agents of malaria, one of the most devastating infectious diseases in human history. Transmitted through the bite of infected female Anopheles mosquitoes, Plasmodium embarks on a complex and insidious life cycle within both its mosquito vector and its human host, a cycle that results in widespread illness and mortality, particularly in tropical and subtropical regions of the world. When an infected mosquito takes a blood meal, it injects Plasmodium sporozoites into the human bloodstream. These tiny, spindle-shaped forms rapidly travel to the liver, where they invade liver cells and begin to multiply asexually, producing thousands of merozoites. This liver stage is typically asymptomatic. After a period of maturation, the liver cells rupture, releasing the merozoites into the bloodstream. It is here in the blood that the most characteristic and damaging phase of malaria begins, as the merozoites invade red blood cells. Inside the red blood cells, the Plasmodium parasites continue to multiply, feeding on hemoglobin and developing through various stages, ring forms, trophozoites and schizonts. Eventually, the infected red blood cells burst, releasing a new generation of merozoites to infect more red blood cells, and also releasing toxins that trigger the hallmark symptoms of malaria, recurring cycles of fever, chills, sweating, and body aches. This cyclical destruction of red blood cells can lead to severe anemia, organ damage, and, in the case of Plasmodium falciparum, the most virulent species, life-threatening complications such as cerebral malaria. The creepy aspect of Plasmodium is multifaceted. There is the stealth of its initial liver invasion, the relentless cyclical destruction it wreaks upon our blood cells, and the sheer scale of its global impact. Visualizing these parasites multiplying within the very cells responsible for carrying oxygen throughout our bodies is a deeply unsettling thought. The mosquito, often a mere nuisance, transforms into a deadly vector, a tiny syringe, delivering a microscopic payload capable of causing immense suffering. The parasite's ability to evade the host's immune system through various sophisticated mechanisms adds to its formidable nature. Some merozoites, instead of continuing the asexual cycle, develop into male and female gametocytes within the red blood cells. When another Anopheles mosquito bites an infected person, it ingests these gametocytes. Inside the mosquito's gut, the gametocytes mature into gametes, fertilization occurs, and a new cycle of development begins, ultimately producing sporozoites that migrate to the mosquito's salivary glands, ready to infect another human. This intricate dance between human and mosquito hosts highlights the parasite's evolutionary sophistication. Plasmodium remains a profound challenge to global health. a microscopic killer whose intricate biology continues to be a major focus of research.